Welcome to the Global Genetic Consortium, and we have Dr. Shailaja Tibrewal, who's the Secretary General of the Global Genetic Consortium, and she's a consultant ophthalmologist and did her stint in NIH as a ophthalmic geneticist, and she will now start the session by giving an introduction.
Thank you, Professor Natarajan, and thank you all the esteemed panelists, chairpersons, and speakers, and all the audience for coming for this session. I would go with a brief round of introductions, following by the talks, then by our esteemed speakers. Professor S. Natarajan, it's my privilege and honor to introduce him. He's a Padma Shri awardee with over 35 years of experience in ophthalmology. He specializes in vitreous and retinal surgeries and performed in number of surgeries, and he's also a teacher, great teacher, and has trained 68 plus vitreoretinal surgeons, and he's a prolific speaker and has given multiple lectures. He's focused on blindness prevention and advancing vitreoretinal surgery across India and across the globe. Next, we have with us Professor Calvin Pang, who is a senior professor at the Visual Sciences at CUHK and the director of the Shantou International Center. And he's a leader in ophthalmology and visual sciences. He's known for his work in molecular genetics of eye diseases, ocular pharmacokinetics, and myopia prevention. He is also an excellent teacher, delivered over 200 invited lectures worldwide. He has several awards and recognitions, especially the Technology Progress Awards, the APAO Senior Achievement Award, and is also the honorary fellow of the College of Ophthalmologists of Hong Kong. He has over 600 publications on molecular genomics and eye disease models, and also on the impact of COVID-19 on ophthalmic health. Professor Namrata Sharma, we all know, and no need for an introduction for her. Next, we have Dr. Janie Wiggs, Paul Austin. She's the Paul Austin Chandler Professor in Ophthalmology at the Harvard Medical School. She's an expert in medical genetics, especially glaucoma. We have all grown, reading her articles and publications on glaucoma genetics. She has a Lifetime Achievement Award and Mentoring Award from the Harvard Ophthalmology. She also got the Teacher of the Year Award for the Harvard Ophthalmology residents. She has helped mentees secure NIH funding, mentored numerous future ophthalmology leaders, and we are so fortunate to have her on our panel today. And next, we have Dr. Gerald Schultz, who is a surgeon at the Eisenhower Medical Center. He's also the chief of ophthalmology at the Desert Regional Medical Center and associate professor at the Loma Linda University. His focus is on ophthalmic genetics and anterior segment surgery. He has lectured at the AIOS, and he has a special affinity for India, and thank you for that, and teaching us always, periodically coming to India and teaching us. He has gold medal from the Bombay Ophthalmic Association. Professor Anne Slavatinek unfortunately could not join us personally, but she has sent across a recorded talk. She is the Director of Division of Human Genetics at the and Professor of Pediatrics at the University of Cincinnati Hospital, and her focus is on dysmorphology and medical genetics, and especially she specializes in the developmental eye defects and multiple congenital anomaly syndromes. She aims to uncover the genetic causes of disorders to improve the diagnosis and treatment. Her pioneering works in pediatric genetics and advancing the genetic disorders and their clinical management. We have with us Dr. Vinay Gupta, who is a distinguished professor at the Glaucoma Services Department at RP Center, AIMS. And he completed his glaucoma fellowship at the Royal Victoria Eye and Ear Hospital, Melbourne, and holds an MBA from the Delhi University. He specializes in congenital, juvenile, surgical glaucomas and glaucoma genetics, authors 220 plus index publications. He is the associate editor for the Indian Journal of Ophthalmology, on the editorial board of AJ Case Reports and Eye. He's also the founding president of the Indian Pediatric Glaucoma Society, and numerous awards to his name, and the recent Indo-US collaborative grant also. And I think Dr. Natarajan has already introduced me, so that is where I'll end with all my introductions and invite Dr. Calvin Pang for his talk.
By the time Dr. Calvin Pang getting his slides.
That side or this side? His talk. Dr. Pang. Which side? Sir.
This side?
Which side? Here. This side.
You have to go there.
It's called here.
Okay, you got it.
Right, thanks very much. Thank you very much, Soja. I'm sorry, I have to give the first talk and then I have to rush because of another meeting, so I couldn't stay and hear the other speakers. I'm sorry about that. Now, I was asked by Soja, she's in charge of this meeting, to talk something on myopia. But yesterday, my colleague, Professor Gai Chen, in a genetic meeting, had shared some of our data already on myopia genetics. Right? But then I'll mention some, and also I'll go back to the beginning, how we organize this cohort for our genetic studies on myopia. And then lovely to have you around so that hopefully we could have the opportunities for collaboration. Right? So myopia, very, very high prevalence in the world, right? We all know that. And in Hong Kong, extremely high. Unfortunately, we are among the top, if not the top city in the world that has the highest prevalence of myopia across all ages in Hong Kong. So we have good reason to study myopia. So that's how we organize our myopia study. We give ourselves a task on myopia prevention at childhood. So we start our work in this particular work. We do more on childhood myopia than elders, but also, of course, we have to do elders. But still, for the childhood myopia, that's our major attention. We organize ourselves in 3 studies. One is the big one, that is the Hong Kong Children Eye Study. It's a territory-wide across all districts of the whole of Hong Kong, which is a city in itself. The parent, children, and their parents, so family, children, and parents together. We study the risk factors, lifestyle, parental style, and mental health, public education, together with community service. So it's involved in a big territorial study since 2015. I'll come back to that later. And out of this study, we set aside the Hong Kong Children Eye Genetic Studies to establish the biobanks for children and their parents, right? So for this, we conducted GWAS and whole exome sequencing materials, genotyping myopia genes. So this is for genetics, the Hong Kong Children Eye Genetic Study, which is part of the Hong Kong Children Eye Study. But then these are children, families that we have children and parents with complete ophthalmic data. And out of this, we also organized the low concentration atropine for myopia progression study, so-called the LAM study, that we utilize 0.05%, 0.05%, beg your pardon, of atropine, low concentration, and then we have conducted randomized controlled trial and with placebo for this atropine study. But we also have a genetic dimension in this study. So the study cohorts, how we organize it, we have so-called patients and controls, right? However, it's still the whole of Hong Kong. We recruit without any selection people that could come to us as a territory-wide population study. Recruitment study subjects were patients and controls, but then the patients are from the children, controls also from these children, and then we do association studies, we do examinations. Now, for some patients, we would have complete imaging examinations. Some, for example, the thyroid eye dissipation, we have endocrinology. But all in all, we have general demographic information, complete ophthalmic investigations, and then we collect all the necessary data. So that's the study itself. We started in 2015 up to today, it's still going on, at the CHK Eye Center based in the Hong Kong Eye Hospital. And we haven't reached 50,000 yet. It's 48,000 something, right? It's going on. Our aim is to complete over 50,000 families. That means we have at least 50,000 trials, i.e. a child. Now, not necessarily 50,000 trials, right? That we may aim at that, but then of course, we have parents that are also myopic, so we can't form trials on that. So that's a bit of a misnomer. But still, we will have 50,000 children between 8 to 10 years old, and actually now we have expanded it to 3 years old. And both of their parents, so we all have their parents together. It's territory-wide among all the districts in Hong Kong. So on every Saturday from 9 a.m. to 6 p.m., we could invited 100 families, right, each Saturday to come into our eye center and to do complete ophthalmic investigation. We take blood and things like that. And since 2022, we extend our study to 3 to 6 years old children, and also special education need children and ethnic minorities. So we do that, and so they, instead of going out to recruit them, we invited them in so that they will have complete ophthalmic investigations in the clinic, right, as if they were patients. And of course, we collect blood from children and parents. And also we collect buccal swab if we can't take blood, but essentially we take blood for DNA. And we also work on the conjunctival swab for ocular microbiome, which is another study. So for the lifestyle, we give out detailed questionnaires before they came. I emailed them or by text. And then they came back. Then all these questionnaires, fill in the details, 350 questions are all assisted. So we train medical students to go through the questionnaires with the parent to standardize, the information, right? So we do that, and also it's part of education to the parents. So we do that. So each family will spend about 45 minutes, a face-to-face interview, and complete these questionnaires while they are with us, while the child and the parents receive ophthalmic investigation. That's what we do. And in the meantime, because we need to have people coming in, so we have strong community, or what I would say heavy community engagement in the TV, and then public education. We go to the mall. Go to the malls, we still do that, and also we go out, and then it's more on publicity, right? That's what we do all over the city of Hong Kong. So that's what we've been doing. Even today, we're still continuing until we reach the goal of over 50,000, right? So on the genetic part, so colloquially, we'll say it's genomics or scientifically, or refractive error in general, myopia based on spherical equivalent and axial length cycloplegic refractive errors, and then endophenotype, and also we also follow this every 3 years. So it's also myopic progression, right? And also part of it, we do the atropine treatment response. I always have to mention, we always mention this first paper that we published. This is actually 2,055 trials, right? But then, of course, we have other families. I think this is not trials because some parents are myopic, so it is wrong. It should be 2,055 families, right? Parental myopia. So we can see that very clearly that there's a dosage effect, right? So when both parents are highly myopic, the children have a high likelihood of myopia. So this is what we have done. And then we went on to do some genotyping with axial length, and also we have some children have 3-year follow-up. So all in all, we have done these genotype studies. So I published all this, but then we went on to do genomics, and then we conducted GWAS of these children. Some have received atropine, and some have responded. So we have done this, and then the data have been out, and now we are working on replication. So it's not published yet. So we haven't published this data, and we have data in the longitudinal studies, 3-year data, 1,000 study subjects. So we have this needs, and now we are doing the validation in another cohort. So that's what we've been doing. Also ocular trace, corneal astigmatism, and the corneal curvature, and all this detailed endophenotype. We also did meta-analysis, GWAS for that, and the myopia and trait, and also the genomics for atropine treatment, the low dosage atropine, the responses. The study was on 262 children. And then we have the treatment response, and we can combine meta-analysis. So we have all these needs now, and then we are organizing a study for the atropine treatment for validation, right? And we have the treatment responses. So we have conducted a randomized control trial published 2 years ago, and now we are replicating this genetic GWAS list in the LAM2. This another stage of our another phase of our LAM studies, right? And the number of study subjects went up to 400. So we're doing that. So what our conclusion is we have so far have accumulated some SNPs with significantly associated children baseline equivalent, and then we have association baseline AL, axial length, and chronic curvature, and there are suggested associations with some multiple SNPs, multiple genes, but they are all under validation now. So hopefully, give us another several months, we'll complete that, and we should be very glad to publish some of our findings. So I acknowledge the team that in Hong Kong in the department that work on this, and then we do need not just validation, go on to do deeper. So I've called for collaboration, all right? And welcome to Hong Kong in the next APAO in Hong Kong, and this is Hong Kong at night. So thank you very much. Thank you.
Thank you, Dr. Kelvin. You have some, yeah.
All right, I have one question, and that is the use of cell phones, tablets, children now reading, they even read books, believe it or not, and I see it more in the Asian children that have a higher incidence of myopia. So I'm wondering if the usage of their eyes at near is also a contributing factor, and this also may spread to other areas of the world. There's sponsorship in India at the Prasad, I think at Rao's Hospital that sponsors studies in myopia.
Thanks very much. Very important question. And this is what we call near work, right? Whether it's tablets or screen and books, right? So we have data on that now because of these questionnaires. And actually, in one study, we also have sensors, right, that can measure the reading distance. So we have information on that, but it is very clear from what have been published and some of our recent data. Yes, definitely near work is associated with childhood myopia. Right? That's definitely. The other, of course, is outdoor time as well. Yeah, you're right. Thank you.
Any other comments, Dr. Kumar? Dr. Vinay, yeah.
If there are patients who have a genetic predisposition to myopia, would atropine treatment help in such patients?
It should. It should. But of course, we are going to have all the information, right? So from our atropine study cohort, of course, we have the phenotype and then the responses of treatment, and of course, then we will genotype all the known related genes or SNPs. Yeah. And regardless of the genotyping, they should have response. But then the response may have, association with some genotype. So again, we will going to find it out later. Thanks very much.
So Kelvin, I think when they say that you should play 1 to 2 hours in the sunlight during the sunset. Maybe Kumar, me, and Jenny played a lot, so we don't wear glasses.
Right.
Okay. So Kumar and me are classmates, and we don't wear glasses. All our classmates wonder how come.
Well, actually, I wear glasses just to see.
All right. Okay. Thanks very much.
Thank you, Kelvin, for coming all the way. Thank you. So next we have a John Prakash recorded talk. No. Then Dr. Musa Khan. No. Then? So Dr. Schultz next. And Dr. Jenny is also then. Okay. So next is we have Dr. Gerald Schultz. We call him Jerry. He is coming to All India Ophthalmic from 1983, I think. And I remember at 83, I came for a meeting when I was a postgraduate, and almost 40 years he is attending almost every year. So Jerry is a All India Ophthalmic Society honorary member.
Yeah, there's a personal interest that I have in ophthalmic genetics because members of my family, we have congenital cataracts inherited in my family. Actually, it's my wife's family. And also I have patient lines with various genetic conditions like microcornea. Other conditions too, and glaucoma we study. So this has personal meaning. And yet on the airplane coming here from the United States, the woman sitting right next to me, her child has retinitis pigmentosa. How do I know this? Because she saw me with my laptop going over my talk, and that started a whole conversation. And I happened to have the literature on that in my computer, so she was studying the umpteenth amount of literature on her child's condition while we're flying over here. So it's very personal. So let's see. We are going to take a look at where we are today in genetic treatments. Of course, I have no financial interest in any of the manufacture of genes. Here's a list of the various genes that can cause ophthalmic disorders. They're huge. And it turns out that about 90% of conditions that affect the body will have some representation in the eyes. Now we've got to take a look. There's also an elephant in the room. It's hard to ignore that. Now, let's go into some definition. Genetics, of course, is the study of specific genes and their DNA and RNA for inheritance, et cetera, for specific therapy for them. But genomics is the study of all the body's genes, the whole genome sequencing. I just want to mention that because ophthalmology, as I showed you, is part of genomics. Gene sequences, for those realize it's only a tiny portion in a chromosome, and for RPE65 it happens to be chromosome 1, and then spot is number 1. There are a lot of new technologies for determining where the genes are precisely, and that's very important. Now, this is at Cornell. They put in this system, so this is for the study of genomics. And where I am in San Diego, we have the Singlet Genomic Sequencer that very precisely, very quickly could analyze and spot the DNA and the genes in the whole body. So that's not an issue right now because we have that. Now the issue is how we're going to treat the problems and what vectors we have that can go straight to the offending mutation and correct it. The home testing kits, here's a list of them. Oh, by the way, 23andMe is now bankrupt and out of business. Again, I'm showing you all the gene variants that affect the eyes. And with that, let's take a look at the naming of it. The Hugo Gene Nomenclature Committee has set up names for the known human genes. They've named about 19,000 of them. There's still about 25,000. And the reason is that a lot of researchers put their own names on the genes that they're working on, but the Hugo is trying to stabilize it. Now, Craig Venter is in my area. It says, yes, he was in Boston, but his plant is on the campus of University of California, San Diego. And I mention him because he has worked on with the idea that you change the DNA software, change the species. It's very much like the computer binary code. You change some of the coding, and of course you get different codings for whatever you're working on. Now here's some of the options for treatment. We have the viral vectors and its associates, adeno-associated virus, and the non-viral strategies. Ribosomes, antisense, suicide gene therapy, and CRISPR, which I'll talk about a little bit more. So the adenoviruses are non-enveloped, double-stranded DNA viruses that can infect a broad range of human and non-human cell types. And they could be used for genetic treatment. And then we have the encapsulated variation. And the most popular one is the adeno-associated virus vectors because they could actually penetrate the membranes of the nucleus and get to the DNA. There's the non-viral vectors. We've got the growth factor gene therapy to enhance the survival. This is for retinal photoreceptors. And we have the anti-apoptosis gene therapy so that the tissue does not die. It slows down the death of the cells of the proteins for the photoreceptors. Then we have the suicide gene therapy to knock out totally an offending gene or DNA. And then the HSV transduce cells. This is, believe it or not, with the medication ganciclovir, which I use quite a bit, because this can also affect the genes to keep them from the herpes simplex and the alterations that virus can cause. So here's some gene therapy, the vectors that can be transmitted. And I did mention the AAV, and the most popular one is 2. It's most workable. And that one, it's used for the inherited retinal dystrophies. I did mention the biallelic RPE65 mutations on the short arm of chromosome 1, and it's used for retinitis pigmentosa. It's also affecting LCA and the fundus alipunctus. So this is how it works. The gene is placed in the AAV2, and that is then breaks through the membranes. This little fellow will then enter the nucleus and change whatever it has to and cause the protein. And the method, of course, is to transmit it intravitreal, but also subretinal. It's not always working. This is being worked on. So the gene delivery does involve placing a strand of DNA, as I mentioned, in a virus that normally enters the human cell and can drop off the gene. We talked about herpes simplex virus. And there's a condition that it's being treated, and that is not the retina, the cornea. And this is being done at Bascom Palmer. It's formulated into an eye drop version, remove the scar of the cornea, and by putting this repair in it, this is done by Dr. Alfonso Sabeda. This solution can replace the genes and clearing of the cornea. It's unbelievable. It's still being worked on. I feel that the coronavirus could be helpful. Watch this. You put the replacement gene in the spike in that, and that could transmit, because we know how coronavirus could affect the human body, everyone's body. But let's put it to use. It could break through the membranes with the repair gene and hopefully reverse it. Then we have non-viral strategies. Just give me a minute. The ribosomes, the antisense, oligonucleotides, etc. I want to mention CRISPR, because Jennifer Doudna won the Nobel Prize for this. Here's what CRISPR stands for, and what this does with the Cas9, it could cut out segments of the gene of the DNA, repair it, and hopefully get what we want. It's used with the Cas9, that's the enzyme. Of course, this can be controversial if doing it to human embryos and therefore making people that we want. Here's other methods: electroporation, high-intensity electro fields, and iontophoresis, low voltage that could change the current. Space flight, yes, it's in the papers. Ionizing space radiation and cellular DNA alterations, not only in the whole body, but also in the cornea. And our space people who were up there for, what, almost a year? Believe me, they're being studied for this, because it's been shown in rats that were in space for quite a while. Now, I mentioned the elephant in the room, and this is one of my colleagues at Cornell that does work on AI and genetics. And the base programs models really can revolutionize how we think about biology and how we develop it. And there's a lot of AI papers that show about the DNA sequences. But in the Eye journal, a few months ago, there's a warning about it, the modern threats in academia. And I discussed this with my professors at Cornell about the papers that are being put out by the students and fellows and how much was done with AI and how much from the brain. So anyway, here's my references. And we're off to the future. Thank you.
Thank you, Jerry. Anybody has any questions or comments?
Thank you, Dr. Schultz, for that excellent talk. Thank you for making it so simplified. But my always, as a clinician, query is that, most of these gene therapies are like targeted therapies for specific genes. Like CRISPR is still a little bit more, like you can use it more widely. But which other therapies do you think would have a more wider application? Like we don't have to depend upon a particular genotype or a particular gene and specifically target those kind of diseases.
Well, this has been bothering me too, because how do we get into the gene that's offending? We know what could be done, but how can we do it and be specific? Now, with the retina, we could put it under the retina and maybe that will change, and hopefully the replacement genes will multiply and knock out the offending genes. Yes, you hit right on the problem.
Kumar, yeah.
The answer to my question is that there is currently a lot of traction on agnostic therapies or non-gene dependent therapies, which are anti-inflammatory in nature or anti-apoptotic in nature. So these are not specific for a gene, and therefore can be easily scaled into a good business model, and therefore can be applied universally. So a patient with what kind of mutation doesn't matter. So they start off with inflammation, then you have oxidative stress, then you have apoptosis. So you try to manage your patient with all these kind of multiple therapies. So that's how the field is moving.
By the way, I want to invite all of you to our session tomorrow morning on ophthalmic genetics. We're going into the basics and expanding it. 8 o'clock in the morning, the basement room A, whatever that name is, the
Amaritas.
tongue twisters, but it's down there, 8 o'clock in the morning.
Amaritas. So I'll invite Dr. Jenny. Dr. Jenny, next you can do it. Yeah. So next we have Dr. Jenny Weeks to speak from Harvard. So you're signing, Jenny. Okay.
I see them here, but I don't see them. Oh, great. Okay, thank you.
Yes, sir.
All right, thanks very much for inviting me to speak in this session. I really appreciate the collaborations that I've had so far, starting really with Kumar 20 years ago here in India, and then some collaborations with Ronnie George at Shankar Nathraya, and now Dr. Vinny Gupta and I are starting a new collaboration here in India, which is very exciting. But for today's talk, I'm going to talk about a couple of collaborations that actually have concluded. These are disclosures which are not relevant. So we all know that glaucoma is a very important disease, common cause of blindness, and that disease screening and risk stratification are challenging. And as a result, many people have glaucoma and don't know it. But genetics can actually help this problem because genes that cause or contribute to glaucoma can be used for genetic testing to identify people who are at risk for disease, and can also be used to identify novel targets for gene-based therapies, something that we were just hearing about a moment ago. To find these genes, it's really been helpful to form these large-scale consortium. And this is true not only in glaucoma, but really across human genetics, consortium have been extremely useful for gene discovery. And there's several reasons for that. First of all, the consortium brings together people with the necessary expertise. So these kinds of genetic studies require somebody who's an expert clinician, who can do phenotyping, requires a team to do molecular biology and bioinformatics, and requires another team, statistical team, who can do the statistical genetics. Another feature of having collaboration in consortium is that it allows for larger sample sizes, which is very important. I'll show you for many studies. It increases the power of our associations. And then also, these kinds of collaborations can facilitate the inclusion of understudied population and groups, which I'll show you also in a minute. So I'm going to give you two examples today. One from early onset glaucoma genes, so the kinds of genes that affect children, and the second from primary open-angle glaucoma, which affects adults. So first, the children. In the US population, I know this would be different here in India or in other populations, but in the US population, the genes that are known to cause early onset glaucoma only account for disease in about 20% of people. So that's wonderful for those 20% of people. We can offer excellent genetic testing, but for many people that come to us for genetic testing, we can't give them useful information. One of the reasons, or one of the concerns that we thought that was limiting these results is that many of these studies have been carried out in US and white Caucasian populations. So we thought it would be interesting and useful to expand this kind of genetic discovery to ethnically diverse populations. So one population that we had the opportunity to study a few years ago was from the Philippines. And this is a picture of Ryan Calantes, who maybe some of you know, who came to my lab and worked for 5 years on a collection of families that he brought from the Philippines. These are families all with juvenile open-angle glaucoma. And we did a sort of a traditional approach to gene discovery where we did whole exome sequencing, looked for mutation in known genes, found some families with mycilin, and then looked for new genes. And interestingly, we found that 3 of these families had mutations in a gene called EFEMP1. And these are the 3 families. You can see 2 of them are really quite large and 1 smaller family. So these variants that we found in EFEMP1 were all highly evolutionarily conserved and quite likely to be the cause of disease in these families. Now, you may have heard about this gene before. This is also called fibulin 3, and this is a gene that causes Duane honeycomb retinal dystrophy. A particular missense allele, one missense mutation, causes Duane's. There was also a family a number of years ago with a rare EFEMP1 mutation, a POAG family, and common variants in this gene have been associated with IOP and POAG and GWAS, but this was the first report of EFEMP1 mutations in early onset glaucoma. And we think that we see this because this is something that exists in the Filipino population and not in the US population. We showed that these mutations cause intracellular protein retention, a functional study, and we have a genotype-phenotype correlation that we think explains the difference between why you would get glaucoma or Duane's if you have mutations in this gene. But I'm showing this here really to emphasize the point that we would not have found this result if we had not studied the families from the Philippines. Now, the second example I want to give you is primary open-angle glaucoma. And to find genes for this, we first formed the Neighborhood Consortium, which included sites, again, all over the United States, and included ophthalmologists, geneticists, bioinformaticists. This was a very successful collaboration. We identified 3 new loci for POAG, and we're fortunate to publish this in Nature Genetics. But at this point, it's still explained just a small percentage of heritability for POAG. So we expanded this to form the International Glaucoma Genetics Consortium. And this map of the world shows the names of the cohorts from all over the world that have been included in this collaboration. Putting all of this together, we ended up with many, many cases and controls, 37,000 cases, which gave us really good statistical power to find genetic variants, and also improved our diversity in this study. While we still have many European Caucasians, we were able to include a number of Asians and a number of Africans in this work. Because of this, we ended up not finding just 3 primary open-angle glaucoma loci, but 127, and expanding across the genome, so greatly expanded the number of loci that we found and the significance of the work. Also, this very large sample size allowed us to do a number of other kinds of analyses. We could do pathway studies to see like which pathways are important in primary open-angle glaucoma, things like laminar fluid shear stress and lipid transport and so on. Interestingly, we were also able to look at the difference in association effects between different ethnic groups, so comparing Caucasians to Africans and comparing Caucasians to Asians, which that also gave us some really useful information that we would not have been able to do if we hadn't included these other groups in our study. The International Glaucoma Genetics Consortium has been incredibly successful, both before and after we completed that GWAS. We have more than 30 publications from the consortium. It's supported many collaborations. Just kind of the nuts and bolts of it, we have monthly meetings and an annual in-person meeting at ARVO. But forming this consortium really allowed us to make a lot of progress in primary open-angle glaucoma genetics. So to summarize, international collaborations have had really important impact on glaucoma genetics research. It allows for larger and ethnically diverse studies. The example that I gave for that is this International Glaucoma Genetics Consortium Trans-Ancestry Meta-Analysis. And also these kind of collaborations allow us to look at unique populations, for example, the Filipino juvenile open-angle glaucoma and our discovery of EFEMP1 as a causative gene. So I just want to acknowledge some people. This is, first of all, a lot of people from the Philippines who worked hard on that study for EFEMP1, our neighborhood consortium, and then all the people who are in the International Glaucoma Genetics Consortium. Thank you very much.
Thank you, Jenny. Anybody wants? Yes, Kumar.
Thank you. Good talk, Vinny. So the question is about the Fibrillin 3 gene variant in glaucoma. So have you followed them up to see if they have developed any retinal degeneration?
Yeah, that's a really excellent question. And I kind of went over that fast because I didn't want to go to, oh, it looks like it's not here anymore. But it turns out that the variant that causes adult onset POAG, so there's 3 variants. Or 3 classes of variants. There's variants that cause severe early onset glaucoma, like juvenile. There's a single variant that causes adult onset disease, so not severe glaucoma, but adult onset. And then there's the variant that causes Doyne's honeycomb dystrophy. It turns out that the POAG variant, those people have both glaucoma and retinal dystrophy. But at the ends of the extreme, the severe glaucoma people do not have retinal degeneration, and the people with Doyne's honeycomb dystrophy do not have glaucoma. And we think it has to do with how much of the protein is sequestered within the cell. And so the POAG one is intermediate. The early onset glaucomas, they're 100% in the cell. They can't get out. The Doyne's gets out, but it's not normal, so it forms these deposits. The POAG one is in between. It's partly in the cell and partly out, and I think that's why there's both phenotypes there.
Very
Yeah.
interesting.
Yeah, it is very interesting. Thank you. Yeah.
It's excellent talk again. I was wondering how's data sharing happening with the consortium? Because that's a very critical question in most of the collaborations that we have, especially for genomic data and DNA related data.
Yeah, thank you for that question. It's an excellent question. And so for the GWAS, all of those groups that I showed on the world map had collections of cases and controls that they had phenotyped and genotyped. What we did for each site is we asked them to do the analysis. We had an analysis script that we shared among all the sites, and we had each site do their own analysis and then share the summary statistics with us, so that there was no sharing of individual level data, which is usually what the problem is.
That's a good plan.
Yeah, no, it worked really well. And I think going forward, that's a great way to do it as well. Yeah.
I'd also like to thank Dr. Shailaja for organizing this on behalf of the Genetic AI Consortium. So we all know what is the gene therapy, and thanks to the advancements in medical science and technology, particularly the mapping of human genome, researchers have been able to pinpoint specific genes associated with a number of inherited eye disorders of the retina, and these includes the Leber congenital amaurosis, the Usher syndrome, Stargardt's disease, retinitis pigmentosa, excellent retinoschisis. And we have Professor Kumar Manikvel here who worked on the RP65, and which has led to the Luxturna therapy now. And I think thanks to people like Kumar who has worked from India along with the rest of the collaborators. And some eye diseases are caused by a defect in a single gene passed down through family members, and most affect the retina or the choroid. And the abnormal gene keeps certain eye cells from working properly, which can lead to vision problems. And sometimes these problems get worse over time. And that's why Arnold Sosby, in his textbook written Ophthalmology is the Playground for Genetics. And gene therapy works by replacing or inactivating disease-causing genes. In some cases, gene therapy introduces new genes into your body to treat a specific disease. With gene therapy, healthcare providers deliver a healthy copy of a gene to cells inside your body. This healthy gene may replace a damaged or a mutated gene, inactivate a mutated gene, or introduce an entirely new gene. In gene therapy, these faulty genes are replaced with healthy copies, and the working genes are delivered to eye cells in the retina during the same procedure. And the gene addition inserts a new copy of gene into your cells, and gene silencing transfers genetic material that prevents the activity of a gene that's already in a cell. And gene editing modifies parts of your DNA by altering or deleting elements within your gene. Our President of India launches India's first homegrown gene therapy for cancer. Accessible and affordable CAR T cell therapy provides a new hope for the whole of humankind, and that's what is there in the press release which is released recently. And India's population recently surpassed China's, making healthcare a crucial issue for the coming decade. Genetic diseases such as sickle cell anemia, beta thalassemia, and retinal dystrophies are prevalent in India due to consanguinity and founder effects. Therapies such as CRISPR-based gene, which are both the speakers mentioned, editing need to be available in India to reduce the dependence on costly imported drugs, which has been published by Arka Ghosh from Bangalore. In India, healthcare is mainly community-based, and there is little awareness of rare genetic diseases, although this has recently improved. The pandemic demonstrated India's ability to scale up production of biologicals such as vaccines, and there is now governmental and non-governmental support for gene and cell therapy programs, including the sickle cell gene editing mission, the development of chimeric antigen receptor, the CAR T cell therapy, and indigenous vaccine development. And we are approaching a critical inflection point for gene editing technology in India, driven by the convergence of three critical factors: patient awareness, evolution of indigenous technologies, and availability of regulatory approval pathways. And The types of gene therapy, as I mentioned in the beginning, the replacement gene therapy, growth factor which can be added, and then the optogenetics involves genetically altering ganglion cells to become photosensitive. This would be useful in retinal degeneration in which the photoreceptor have already suffered extensive damage. And then gene editing techniques such as CRISPR, they aim to genetically alter or modify the DNA, and the new treatments bring hope to inherited eye diseases. And I think Charles Keepens always mentioned, never say never again to a patient. And I remember one of the first meetings, a public meeting we had in Mumbai that time it was Bombay, in 1991, and he collected along with Dr. Michael Denton for doing genetic study, and all patients were hopeful, but I think he has the patience to explain to the patients about and counseling them, and that way with all the things, and still patients are hopeful. So we are keeping the patient with the hope, but at the same time telling the truth. The promising new treatments are being studied and developed for people with genetic eye disease or retina called gene therapy. This treatment involves replacing non-working genes with healthy copies to slow or halt vision loss. And we are entering an era of new therapy for those who otherwise would become irreversibly blind. For the first time, we are offering hope for this patient and these families and improving quality of life. And Bengaluru-based Narayanathala is developing gene therapy solution that could drastically reduce the cost of treating genetic eye diseases. Currently, gene therapy treatment used to treat about 65 to 70 conditions can cost as high as 8 to 10 crore per dose. And the institute is aiming to cut costs by 70 to 80% and has also received regulatory approval to conduct clinical trials for gene therapy targeting various eye diseases. And I'm happy to mention Dr. Arka along with the Professor Kumar Manikvel and Dr. Rohit Shetty are working on that. The institution also developed a gene repair, regeneration and ophthalmic workstation called the GROW Lab. The GROW Lab is focused on advancing research into gene therapies for several common but challenging eye diseases. Among them are keratoconus, where the cornea becomes thin and cone-shaped, and RP, a degenerative disease that leads to blindness. Additionally, the lab is tackling ARMD, a leading cause of vision loss in elderly population. And this is from the YouTube, the gene therapy breakthrough could cure blindness.
Jonathan Wyatt had given up all hope of ever reading again. He was just 20 when he was told he had a genetic disorder called choroideremia. He would slowly go blind. But for the first time, doctors have used gene therapy to correct a DNA defect inside his eye cells, dramatically improving his vision.
When I got home, I looked at Diana's mobile phone and I said, I can see the digits. I can see the digits. Hey, switch it on. Switch it on, and I said, I can see it.
Can you increase the volume?
The first 6 patients treated at the Oxford Eye Hospital were all able to see better in dim light.
So this is where the surgeon's role comes when the gene therapy is ready. We have to place the gene in the subretinal space. You'll be seeing now they are also trying to use the robot to inject the gene into the submacular space.
Where they inject the virus behind the retina. The virus then infected light-sensitive cells, releasing the gene, which started producing protein that helped to preserve vision.
Rather than taking a pill or proteins or taking tablets, we're actually correcting the disease at the genetic level. In other words, genetically modifying the patients who have the problem to put the gene back that's missing. Obviously, we're very excited because potentially gene replacement is a way of stopping the disease from starting in the first place, and we could genetically modify people to prevent them from getting the disease and hopefully prevent them from losing their sight.
Around 1,000 people in Britain suffer from choroideremia, but the same technique could treat far more common genetic causes of blindness, including age-related macular degeneration. Doctors are so excited by the results published in the Lancet Medical Journal that they've already started another trial. The procedure could be approved.
So there are viral vectors for gene transfer, and the most common method to deliver vectors are intravitreal, but doesn't work much, and that's why the subretinal injection. It's not just a subretinal. You have to do vitreoretinal surgery and go behind the subretinal space, and that's where the skill is required, and as well as I think now the robot they are trying to say so that the injection can be without any tremor, and also fixed amount can be injected into subretinal space. And this video is again from Scientific American, which I borrowed from Professor Kumar a few years back from University College of London.
For sufferers of hereditary blindness, there may be a light at the end of the tunnel. 2 experimental trials have shown that
Ladies.
gene therapy could one day cure a rare form of blindness that affects about 2,000 Americans. The research teams tested the treatment on a total of 6 patients. Here you can see how they performed the eye surgery. Prepared genes were injected into the eye of this young visually impaired patient. The eye was infused with air, then reinfused with fluid near the end of the surgery. To measure the patient's performance before and after the treatment, he was guided through an obstacle course. Here you can see how he navigated through the maze prior to the surgery. He bumped into obstacles 6 times and was disoriented twice. It took him 77 seconds to finish the course. 6 months after the treatment, the patient showed a dramatic improvement. He was able to make his way through the maze in 14 seconds without bumping into anything or getting disoriented. He was one of 4 patients that showed an improvement. The treatment also proved to be safe, which has not been the case with previous gene therapy trials.
So thanks to the new scientific, and there are so many advances taking place, and I think the viruses. So this is the one which RP65 gene mutation, which Dr. Kumar Manigal published in Nature, which finally came for the ocular gene therapy, and so I'll just. Okay, this is another video.
Hyperthermia is a rare genetic disease in which photoreceptors in the retina and underlying vascular layer, known as the
Okay, I think.
The first 6.
So in summary, vision loss from inheritable disease is a debilitating condition. Genotyping is the key to developing possible gene-based therapies, and many gene therapies are under the pipeline and a ray of hope for the patients. And I think thank you very much for the listening. Any comments or yes?
Ever since the National Rare Disease Policy has come along in India and it was revised in 2022 as well, it has a clause that says that if there's an actionable mutation or an actionable genetic disorder, the government will provide support. And as you mentioned in your slides, the costs go up to like 8 to 10 crores. So do you have any real-time example of any case of an eye disorder where a person is actually utilized, or how the government has supported, even if it's something like sending them abroad for a clinical trial or such?
So I think a lot of patients have approached me, but I have not got any patient who has got a permission. Particularly, so many patients we have retinitis pigmentosa. Nobody, 2 patients were ready to go for a trial, but unfortunately, they're not unfortunately, the patient did not have RP65 mutation. They were actually disappointed they don't have. And I think the expectation of patients are also high, but I don't know about this, whether this particular thing which has come in 22, I'm not aware, Kumar, the government is given, but I don't know whether any patient example, I don't have any patient example where they have benefited from this government scheme.
Yes, actually, what was mentioned is correct. There was an incident in Kerala for another genetic disorder, a rare one, I think was a spinal muscular atrophy, SMA disorders, for which there was a collection of money which was done as a crowd source. Okay, and they collected the money, and somebody put a case in the court that how you can collect like that, and then it went into a spin, and the medicine came. Apparently, the child died.
Oh.
Okay. So there is an initiative. For example, Government of Tamil Nadu does support rare diseases, and they are ready to do a diagnostic testing. Therapy still, it is a big question mark because they're very expensive.
Yes.
But I think Government of India is very open about it.
Okay, good, but I don't have any patients. Shailaja, you have any patients? No, where they benefited from government.
No, not from government. One of my patients from achromatopsia approached Morphils for the trial which was going, but I don't think they followed up later on because they shifted probably there and they never haven't come back. But the child with achromatopsia, so parents are open to shifting out of India also to places where these clinical trials are going on, and maybe enroll their children into the trials and await. But in India per se, crowdfunding for an ophthalmic disorder, I'm not aware of.
So Messi did that. So he migrated from Argentina to Spain, and then that's how he became a football player also, and he got funding to support. So I think it's a good thing if the government is doing. Thank you. So next, can I invite Dr. Vinay Gupta to give a talk? And Vinay Gupta, as I said, is the founding president of Indian Pediatric Glaucoma, and he's also part of the International Council of Ophthalmology with us. Thank you, Vinay. I should have your number. No, I lost your number.
Thank you, Shailja, for inviting me for this talk. I'm basically a clinician, but I'm going to talk about our journey in trying to investigate the genetic basis of certain anterior segment disorders. So as we all know, that what we know at present is just the tip of the iceberg. And in order to know more, a lot needs to be done. For example, for a clinician, it is about establishing the correct phenotype, the severity of the phenotype, to be able to get a good pedigree of that particular individual, collecting DNA samples from as many relatives as possible, trying to sequence them, interpreting that data, and then follow up with their penetrance and expression data. And this all requires a lot of time, money, and of course commitment to the cause. But then, of course, the return on investment is huge. I mean, even if you can help one family, I think it's a huge amount of work that has gone to some benefit. So I'm going to show you some cases of when we require genetic testing and when not. For example, this is a 10-year-old who came with a classic posterior embryotoxon, and it was an axon field rigor malformation, and we really didn't require any genetic testing for this patient because invariably it's either a FOXY1 or a PITEX2 mutation. This is another case where it is a classic case of aniridia, and if it is a familial case, probably we do not require much of genetic testing. Then there are these children who have a totally opaque corneas, typically central opacities with a when you do a UBM, you find this concavity in the posterior cornea, which is invariably a Peters anomaly. And again, really not much of genetic testing can help. But then there are certain situations where you need genetic testing to make a diagnosis. For example, in this particular case where we couldn't see a posterior embryotoxon, the iris was absolutely atrophied, and we really didn't know if it was a primary congenital glaucoma or an axon field rigor malformation. So we subjected the patient to a whole exome sequencing, and there was a FOXY1 mutation. But then there was a similar case like this where we find extensive iris atrophy, iris holes, a whirling of the pupil, and then again, you think whether it's an axon field rigor or primary congenital glaucoma. And here in this case, it turned out to be a CYP1B1 mutation. So the phenotype was, in both the first and second case, was almost similar. But of course, the genetic makeup in the two cases was very different. Then there could be situations where you want to provide the appropriate treatment. And this was, for example, a very interesting case when this young boy at 10 years of age came to me with very high intraocular pressures, and he was a high myope, and we thought it was juvenile open-angle glaucoma. There was a corneal haze that he had, which was probably because of the high intraocular pressure. He underwent a both eyes trabeculectomy. 10 years later, after the trabeculectomy, his intraocular pressure rose again. In the right eye, it was like 27 millimeters of mercury, and his axial lens were normal, but his cornea was really thick. Considering the fact that his discs were relatively normal, we did a repeat trabeculectomy because we didn't want him to progress and become blind. Now he got married and he had a child, and he brought this child to me. The child had a very strange looking cornea, and this kind of corneal problem, I mean, I had never seen before, and my cornea colleagues were a little hesitant about what the diagnosis was. So here this child had a borderline corneal diameter. His axial lens were more or less normal. He was a high myope, but his corneal thickness was much more. So this was a time when we thought that a genetic testing is important to find out what this family had. We also did an IOCT of this child. This child was hardly 1 month old when he came to me, and of course the cornea was thickened. So it was not a case of CHED because it's not classical diffuse corneal haze that this child had. But on exome sequencing, it turned out to be a ZEB1 mutation, which is classically associated with the posterior polymorphous corneal dystrophy, and that is what both the father and the child had, and probably it was a mistaken diagnosis where the father was mistaken to be a juvenile open-angle glaucoma when actually it was a case of a corneal dystrophy, and he was subjected to 2 surgeries for glaucoma when it was actually not glaucoma at all. So here, a sequencing, a genetic diagnosis was important to establish what the phenotype was. Then again, there are certain disorders like this, for example, where you have thickened corneas, and in this particular case, the cornea was as thick as 1,000 microns, and this was a classic case of a CHED. But there are a lot of such children who are brought to us when the misdiagnosis between CHED and primary congenital glaucoma is actually a very borderline kind of condition, and some people think that actually both of them are exclusive to each other, that the primary congenital glaucoma is extremely different from a CHED, but then there are certain people who think that there could be glaucomas even associated with CHED. So then another important case I would like to highlight was of a young boy, another young boy who came to us. He had normal looking corneas, but they were slightly smaller but very high intraocular pressure with a high myopia. They don't give any family history of glaucoma, but you can see that there's an advanced glaucomatous optic atrophy in the left eye, for which we did a trabeculectomy. His corneal diameters were slightly smaller, and this child and his parents underwent whole exome sequencing, where we found a COL4A1 mutation, which is not classically associated with glaucomas, but has been found to be associated with posterior embryotoxin and an axon field Rieger malformation like phenotype. They're also associated with cerebral lesions, and it is known that these children, as they grow up, can have microangiopathy and stroke, in which case they require repeated MRIs. So when we went back and we looked at his gonioscopy, there was a subtle prominence of his Schwall baseline, which was actually found out only on an AIOS OCT. So the MRI was normal, but now we went back and looked at the father, because all this time the father was said to have normal eyes and normal vision, but his father also turned out to have high intraocular pressures. He was an ocular hypertensive, and when we examined him carefully, he also had this posterior embryotoxin, which is extremely subtle. So this particular family actually gave us a new diagnosis, a new phenotype that could be associated with juvenile open-angle glaucoma, which is a COL4A1 mutation. Then it's important to screen and prognosticate certain family members, and we already know that there are certain myocilin mutations which are associated with a very severe phenotype compared to others. So when you are looking for a myocilin mutation, there could be very large families that you are looking for, and this particular mutation, which is 367 with arginine, is associated with a very large family with very high penetrance rate. We have found very large families with this particular mutation, and in such cases it's important that you sequence the family members to find as many members as possible with glaucoma. This was another interesting family where both the kids belonged to the same family, had a CYP1B1 homozygous mutation. This was an arginine 390 histidine, which is associated with very severe type of a congenital glaucoma, and I operated both the kids actually. This boy actually came at 7 days of age, and this is 4 years down the line, and you can see that the corneas are clear, whereas the sister actually for some reason didn't come very early. The surgery was delayed, and she did not have very good outcomes. So I'm just trying to put the point that despite the fact that they have the same mutation, they come from the same family, they're operated by the same surgeon, the same technique, the outcomes are very different, primarily because the different times by which the children were brought. So this is not a classical case of a phenotype-genotype correlation, but it is a classical case of a delayed surgery in a child, which can change the totally. So to summarize, it is important for genetics to make a correct diagnosis and decide treatment in some cases. It is also important because we can understand certain systemic association of some diseases which could be routine in a busy OPD. And then of course you can prognosticate certain diseases, especially when large families are involved, but one has to be careful and have some idea of the social concerns where, especially in the type of scenario we are living in, where pinpointing the genetic mutation and from where it has come can have a lot of familial implications. So one has to be very careful with what kind of genetic counseling we'll give to these patients. Thank you.
Thank you, Dr. Vinay. And I think for want of time,
Yeah.
we'll have Dr. Shailaja's talk, and then in case some questions are there for both of you, we can have.
Yeah, thank you to GEGC and Professor Natarajan for giving me this opportunity. So I would be also talking on the similar lines as Dr. Vinay Gupta, and as a clinician pointing out the instances where genetic diagnosis and a genetic implications have helped me in managing my patients. When I joined work as a pediatric ophthalmologist, I was not aware that that genetic disorder is so prevalent among my patients. In fact, every other child I was seeing had some connection with a genetic genetic mechanism that was happening, either direct Mendelian disorder or some predisposition to a genetic condition or genetic situation. So when I went back and read about it, I saw that inherited eye disorders actually constitute more than 50% of legal blindness in children, and thus they have a huge economic and social impact. If you look at the eye, and these are only the monogenic disorders that I have, monogenic or polygenic disorders which I have mentioned, but not the ones where there are multifactorial disorders which also have a genetic connection. So it could be also complex because of chromosomal involvement, then direct DNA deletions, duplications, or even single nucleotide changes, plus and minus systemic associations. So how does a clinician try to apply this to their own practice? So this talk essentially gives us clinical examples of how application of genetics helped in diagnosis, prevention, or management of the disease, how it helped us understand the segregation of the disease in the family or establish a genotype-phenotype correlation, also some clinical examples of limitations of genetic testing and counseling, some avenues for research in genetics for clinicians, and the last part probably I'll skip because it's already been covered by previous two speakers. So I begin with this 26-year-old male who presented with a history of sudden loss, painless, gradual progressive, very slowly progressive, simultaneous bilateral vision loss at 18 years of age. When he came to me, he was 26 years and was still finding answers as what is happening to him. His old records documented bilateral optic neuritis, for which he had received intravenous methylprednisolone at that time. His current vision was 1/60 in both the eyes, and fundus showed bilateral optic atrophy. So what was odd in this case? I could have easily dismissed this patient as a sequelae of bilateral severe optic neuritis, maybe an NMO positive patient. So what was odd? But he didn't typically fit into the typical optic neuritis gender, being male. Also, the slow progression even after treatment was not fitting into optic neuritis, and MRI being normal, although not unknown, but it's still a little odd. So can I do something more? That was the question. So only a simple thing like a family history, which was not taken before in this patient, revealed that there are two other siblings from the maternal side who have been similarly diagnosed as bilateral optic atrophy at a young age, optic neuritis at a young age, and which is very odd for optic neuritis. So definitely this was a hereditary optic atrophy, most commonly LH1 because it's more common, and also maybe X-linked optic atrophy. But what should I do? Should I examine the parents? Unfortunately, the other family members and parents were not present for examination, but we did genetic testing of the mitochondrial gene and did found a gene in the variation in the MTND4 gene, which signifies Leber's hereditary optic neuropathy. So why is it important to make this diagnosis for this particular patient? And taking this example, a genetic diagnosis helps us in prognostication, which would be very different for optic neuritis as compared to hereditary optic atrophies. The management are quite different. Of course, IVMP was really not required in this patient. Had it been diagnosed at the early time, he could have benefited from Idebenone prophylaxis to prevent involvement of the other eye, or maybe help in a little bit recovery also. And genetic counseling is definitely not applicable for an optic neuritis, but it's very relevant in an LH1, and especially the sister of and the other female siblings of all the affected ones need to be counseled for their children being at risk for this disorder. Family history and examination of parents is extremely important for all suspected genetic diseases. It can sometimes directly clinch a diagnosis. For example, a tapetoretinal sheen in female carriers of X-linked RP, posterior vise sutures in female carriers of Nance-Horan syndrome, and anterior sutures, sutural cataracts in carriers of cerebrotendinous xanthomatosis. So these can directly clinch a diagnosis and even not require a genetic testing. Coming to the second case where the diagnosis was not very difficult. This was a 2-year-old male, observed lack of eye contact at 4 months of age, normal development milestones. There was a hyperopia with nystagmus, ocular digital sign was positive, and a normal fundus. So this was typical Leber's congenital amaurosis. But here the mother was pregnant at this time, and they wanted to know the recurrence risk and also what is the chance of this baby having the disease, and wanted to prevent it. And he was clearly born of a consanguineous marriage. So we told them there's a 25% chance, but they wanted to test it. So at this point, genetic testing helped us to plan a prenatal diagnosis, and a similar mutation was found in the ongoing pregnancy, and the parents, plan to terminate the pregnancy. So here it lies in our, armamentarium. It's our duty to advise them for these kind of prevention strategies which are present. It is very difficult to live with one child with a debilitating and a blinding disorder, and imagine having 2 or 3 kids with the same disorder. So it's a duty to advise them to have this kind of a counseling done, and options of prenatal and pre-implantation genetics all should be given to the parents. And these are some examples of how phenotype-genotype correlation helps, especially in the retinal dystrophies. I'll not go into the details of this. Coming on to the next case, which was a 5-year-old male. This one also had a very similar kind of presentation, and here we suspected that this was a severe early onset rod cone dystrophy, and because of that he also had a hearing issue, probably an Usher syndrome. Here the diagnosis was just made because the parents wanted a genetic testing was done because they wanted a definitive answer. But lo and behold, what we found that he actually had NCL, which is neuronal ceroid lipofuscinosis, and what we were thinking that he's probably having a walking difficulty because of his vision problems actually turned out to be a CNS involvement of this patient, and unfortunately this patient succumbed a couple of months after the diagnosis, in fact. So again, it's the onus of the ophthalmologist sometimes to pick up these disorders and refer them so that these kind of things may not happen if the condition allows. So sometimes you just don't have any other things, but these red flags like any intellectual disabilities, any other neurological issues, some congenital anomalies, dysmorphic features, history of immunodeficiencies, history of recurrent pregnancy loss in the mother, and anything odd should clinch us towards some other problem going on in the child and not dismiss them just as an ocular disease. Coming to one more case where this 11-month-old baby girl, she presented to us with bilaterally small eyes since birth with nystagmus and very poor vision, born of non-consanguineous marriage, the first child of the parents, uneventful pregnancy, no developmental delay. So looking at the external picture, looked very typical of a microcornea with a coloboma, this almond-shaped cornea and pupils, but a deeper look revealed that she also had a PHPV kind of a thing where elongated ciliary process was there. The lens appeared to be inferiorly dislocated and also dislocated posteriorly into the vitreous cavity. I had never seen something like this. I also did a B scan, and here there was a PFV stalk, or the persistent fetal vasculature, there along with the coloboma. So this was a combination of microcornea, posterior megalolenticonus, a persistent fetal vasculature, and chororetinal coloboma. And I had seen this for the first time when I Googled it. It fitted into this MPPC syndrome, and very few cases have been described worldwide, and it has been postulated that somehow that incomplete closure of the embryonic fissure also leads to the non-absorption of the PFV stalk and leads to a combination of this. But it's so rare that there has to be something going on wrong, some other gene interactions having and affecting both these mechanisms or both these developmental processes. But unfortunately, till now, no genetic cause had been identified. Now, these parents were wanting again a diagnosis because they were planning next pregnancy, and they wanted if we could help them. So we did a clinical exome sequencing, did not come out significant for this particular disease, but came out for Smith-Lemli-Epuit syndrome, which the child didn't fit into. So we also did a micro-air and extended micro-air. Again, no testing, but nothing came positive. The mother became pregnant in this process, but we did advise him for a good level 2 ultrasound to at least detect globe abnormalities. So the learning points is that genetic tool may not always provide answers. The disease phenotype offered determines the yield of the tests, and choosing an appropriate testing tool is important. And extremely rare disorders may require research level testing, like in this patient probably a whole genome sequencing would be the next step. It lies in again a duty to report some of these abnormal features or some unusual features that we see in our patients, like in this patient who presented with a diagnosis of Holt-Oram syndrome, referred by a cardiologist, having microphthalmos cataract, but this axon-field rigor like anterior segment dysgenesis didn't fit in the picture, and genetic testing revealed a B-core gene mutation, and this anterior segment dysgenesis had not been reported previously in B-core gene as well. And while we were publishing this case, only one other case got published, so there is a two cases reported to have this anterior segment dysgenesis. So we as clinicians can actually expand the phenotypic, expanse of the phenotypic variety or the phenotypic knowledge of these disorders and have better clinico-genetic correlations. This patient had aniridia, again came to us for prenatal counseling, came out to be a PAX6 mutation, but because of incomplete evidence had a VUS. So segregation analysis in the family confirmed that this is probably a pathogenic mutation, and again reporting this case can help us convert these VUS into likely pathogenic or pathogenic in this. And the last one is, as Dr. Wiggs also mentioned in a talk, is that by collaborations with scientists and basic researchers, we can actually kind of expand these and have a meaningful collaboration, meaningful studies, like with my collaboration with Dr. Ansel Watinek from Cincinnati and Dr. Brooks and Dr. Vanita from Amritsar. We are trying to study the genetic constitution and the differences between 200 patients in India versus the USA, and we'll be doing gene dephenotyping, exome sequencing, and whole genome sequencing, and also in evaluating zebrafish MAC models, especially are targeting the retinoic acid pathway genes, because we believe that probably there are some connection between the maternal vitamin A deficiencies and the retinoic acid pathway genes, which is kind of making the patients in our population have more severe variety of microphthalmos, anophthalmos, and coloboma. So I'll skip this treatment options and in the end to just summarize that supportive care is very much important for these patients. If at all we can't do anything, at least get them a good genetic counseling, link them to various support groups, help them with whatever vision rehabilitation is possible or cosmetic rehabilitation sometimes, and we really need to collaborate with pediatricians, geneticists, genetic counselors, and definitely not send out the patient saying that if there's no cure, it does not mean no hope. So thank you everyone for your patient listening.
Thank you, Dr. Shailaja. Anybody wants to say something? I know we are already crossed the time, but anybody want to have any question? Kumar, you want to say something?
I just wanted to congratulate Shailaja. It's nice to see clinicians talking genetics. So congratulations. Good. You should actually collect more cases and publish more. Thank you.
So I asked Dr. We need to come here and we can have Kumar, you also can join us, and can we have a picture? Maybe you also can come. Thanks for waiting. You also can join us. So thanks for, I think, patiently waiting even after the program is over. Thank you very much. Give your camera, no? Okay. You want us? Oh, come. Okay.
Just on the edge.